Hello everyone. In today's session, we will cover various areas of supervised machine learning. They're called decision trees, random forest, and we'll also introduce you to many other terms and methods that can be used to create predictive models. Before we go anywhere further, let's review some of the aspects of machine learning that we used so far. So, uh, we were dealing mostly with unlabeled data before. Now, data being labeled or unlabeled might be confusing at start because in some general terms, labeled data is just something that has a label, right? But as we said in previous sessions, labeled data in a statistical form is data that is categorized. So we create categories out of genes or samples, for example, and then we are inputting those categories in the analysis. On the other hand, we could have labeled sample like the one we see on the screen, but it's not labeled statistically. Why? For example, we have some labels like normal, for example, in each sample, but that is just part of its ID, of its ID. So we have unique ID, for example, I don't know, first one SRR 204 033944, and then we have dash normal, right? So it's just part of the name. Program or machine learning code won't actually consider this as a category. It's just a name. But we use these names in unsupervised machine learning just to get some input for ourselves to see how did we categorize the data using machine learning or unsupervised methods we used before. You could have a different situation where, for example, a data set is not labeled like this. It's not labeled just in some general term. It could be labeled statistically. It could have another column that actually separates parts of your data. Let me show you how such a data set looks like. So this is the same data set. It's the same data set we used before. It's a data set on that DCIS breast cancer precursors. And this is unlabeled form, statistically unlabeled, and this is labeled form. You can see that it actually has just another column that is marked as class. So in this column, we have non-unique names. What does that mean? It means that we have categories that are identical to each other, but different from some other categories. For example, we have a normal category. So each one of them is labeled equally, just normal, 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 etc. And same goes for basal, luminal B, or any. That is quite different to the situation we had here. Here we had all those labels like normal and every other, but we also had these unique identifiers in the name. So that is one very important difference that actually differentiates two types of data sets uh, that you will have when dealing with unsupervised or supervised machine learning. So unlabeled data, statistically speaking, is the one that has unique identifiers for each of the samples or each of the genes. You don't have same identical labels anywhere. So why is this important? Some of you actually mentioned this issue to me when you tried to use this identically named data sets, that you actually encountered problems with k-means, for example, and then you saw that after using this form with unique identifiers, that problem is solved. Why? Because a lot of unsupervised methods will have assumption. It should be a general assumption. Some of them have it in a rigorous form, some of them don't, but there is an assumption that you actually don't have categories in the data. And the code itself, it could be written in many coding languages, is generally written to recognize only data sets that have unique identifiers because you're using it for unsupervised learning. That is why it's important to differentiate between these two types of data sets. So this is the data set we used before, and this is the data set that has non-unique identifiers. It has same categories across this column class. This is a type of data set that you would like to use when dealing with labeled data, when using supervised machine learning to do another type of analysis. We'll see how it's different from the machine learning method we used before. But for now, I want you to remember this very important information. You need to have a column that actually has those categories, and they're not unique, right? All samples have a label that is identical to all other labels from the same category. For example, normal or basic. Let's get back to some analysis that we did on our last session. So we had unlabeled data. We said that those names are just for us. And machine learning algorithm called word method from hierarchical clustering part of the pipeline actually created this output for us. So in essence, we had all those samples and 22,000 genes, and in the end, we created some categorization, right? We were able to cluster the data in a specific way and find different subgroups in the data. So this is called clustering, as we said, and those clusters are associated in a way that all objects in the same cluster are related, statistically speaking. Very often, this relation is also transferred to biological relation as well. So we could find different subgroups in the data, different subtypes of DCIS, for example. And we could see this hierarchy, branching and everything, but it was all associated to clustering, as we said. Clustering was unsupervised method that was transferred to this hierarchy. Very important thing is that unsupervised methods could be used to create categories in the data. So I could just take these categories and place them in that class column. But for the simplicity of the analysis, we took the categories from the publication itself. And that is also another way that you can do supervised machine learning. You could derive categories based on the information that you have from the clinical point of view. And very often, this is the form of analysis that you will be dealing with when performing machine learning or supervised machine learning methods. So that is quite different from the types of the analysis we used before, because before we had data, right? It's data, and we are deriving categories from the data. Most of our information comes from data. Pretty much everything that we create, all data that we create is derived from the data set. But you could also have external inputs. In this case, we have an external input. We have diagnostically created categories. This that is under class. So we have an external input. And what if we wanted to see what part of this genome or what genes are associated with that external input, with those categories? We would need to create some predictive model to see if some of these categories could be predicted based on some genes. We said when analyzing correlations that if we can predict samples or genes based on each other, that means they're associated. So same principle goes here. If you can predict a class based on one gene, that means that this classification, this external input, is associated with that gene. So let's stop here with all the explanations about genes, labeled, unlabeled data, and everything else, and we'll get back to this during the session. How would we create a predictive model? How would we predict something? For example, predict if any of those classes are related to some genes. We have 22,000 genes, so it's very large number of genes, very large number of predictions you have to make, and very large number of models you have to create and then compare all of them. In the end, you would get some information. So it's huge amount of computing, but luckily machine learning algorithms are programmed to do all of these in a few steps, right? So when we were talking about machine learning algorithms, we said they have large number of steps and then they're repeated to learn something from data and derive results from it. So all of that happens within the algorithm, within that learning process. So we don't have to worry about it at this point too much. So what I want you to remember in this moment is that algorithms should choose best features or predictors for certain subtype of DCIS cancer. How would it do that? Well, simply it would analyze every feature, see how they behave as predictors, and then choose the best. For now, that's a very simplistic explanation about it. So let's consider this. Imagine you have a gene, for example, which is a variant, right? So gene expression across different samples. And you want to use that gene to see if it's predictor for, I don't know, a luminal subtype. So you could use a very simplistic form to, for example, create some, I don't know, initial model. Is that gene expressed in luminal or is it not expressed? If it is expressed, that means it is going towards that branch that could identify luminal subtype. If it's not expressed, that means that it's probably not luminal. But on the other hand, this could branch even further. So we could create another decision. If another gene is expressed along with the first one we used, then we can say, hey, let's create another decision. If it's expressed, that means that we could create some prediction or make a decision that it probably belongs to luminal subtype. So we could have multiple levels, multiple decisions to make before making final prediction. As you can see, this is decision-based model. So these decisions could be made in a simple way, as I said, just by some gene being or not being expressed, but generally in biological samples, as you already saw, well, when analyzing gene expressions, very often we have a situation that some gene is more or less expressed, right? We don't have just zero and maximum, right? So to be more accurate and find relations between levels of gene expressions and those classes, we need to create some cutoff values. So those cutoff values are also analyzed. Each gene is analyzed as a predictor, but also all cutoff values going from 0 to maximum are also analyzed to see if they have predictive potential. So it's very similar to the situation we had before. Just instead of using yes or no, we are using some cutoff value, some decision boundary, and then we look, is the expression of that gene lower or higher than that decision boundary? And that would correspond to this yes and no. So, for example, if it's lower, we go to the left branch, and if it's higher, we go to the right branch or vice versa, depending on the model. So you can also create these decision branches and have a different cutoff value for each of them. In the end, you would create a predictive model like this. So let's analyze the structure of this decision model. We have some initial variable which is called a root variable, and then that root variable is assigned with the first decision point. We have a cutoff value, and then we are deciding if the value of gene expression is higher or lower. And then we have two branches, and both of those branches have their own cutoff value. And then based on the fact that gene expression is lower or higher on them, we are also making some decisions. And in the end, we are creating some predictions. So this model is similar to a tree model. So we have root variable, and then we have branching, and in the end, we have predictions. These predictions are actually called leaf structures of decision tree. The whole model is called decision tree, and predictions are leaf structures. Why are they called leaves? Well, because they don't branch anymore, right? So all other parts of the tree have some branches that go towards them, and then they're branching into smaller branches. But predictions or leaf parts of decision tree, they are the endpoint. They don't branch anymore, and that's why they're called leaves. Before we go any further, let me just give you some brief introduction on how do we actually make these decisions. How do we find these decision boundaries? Well, there are different methods, but the one that we will use today, and most of them are very similar, they all pretty much deal with probability. So you want to see what is the highest probability that you will actually predict something based on some gene. So if genes have small probability of predicting something, that means that they're not good predictors and they won't be chosen by the algorithm. But if you can predict something with high probability, let's say, I don't know, higher than 90%, that would probably be chosen by the algorithm to be one of these variables that will be included in the decision tree. We also have another term here, which is called Gini. In these semicolons, you have inequality and the fact that best predictors have low Gini. So what is Gini? Gini is actually a probability that you will have a bad predictor. It's related to the probability of having predicted. How it's calculated? It's calculated by creating that trend line. We were talking about trend lines when analyzing correlations, and then you can see how much does the line of prediction deviate from that trend line. It's similar to errors, right? So this is not just deviation or standard error, but it's very similar to it. And you can see how much does that curve that is called Lorentz curve. Lorentz curve is a curve that tells you how unequal is your prediction line. So if it's unequal, that means you have bad predictor. If it's completely equal to that trend line, that means you have a very good predictor and your Gini value would be 0. So the higher the Gini or inequality of prediction, the worse predictor you have. Generally, the whole point of creating a decision tree is to find predictors that have lowest Gini. So this is one of the features that is used to select the genes. Also, when selecting cutoff values, you analyze all cutoff values for each predictor, and then you find the cutoff that has also lowest Gini. So this is one of the models that is used to create, for example, decision trees. There are also other models, but generally all of them are related to probability of having good predictor. So this is how a basic decision tree would look like. And we will get here from the analysis of that data set. So it's supervised. We used those classes that were externally inputted. We have information from medical area, from diagnostic area, and we want to find out which genes could be associated with those categories. In other words, which genes could be associated with those categories. So you can see in this case that uh, every decision point has the name of the gene. So that gene is used to create a decision. We also have some value. For example, take a look at this first one, ENSG that ends with 195.14. It says less than 0.34. What that means is that 0.34 is the cutoff value. So if the expression of this gene is lower than 0.34, we go to the left branch. And then we come across another gene, and it has a cutoff value of 0.135. So now, if the value of that gene is higher than 0.135, then we go to the right branch, and we come to luminal A. So here's a simple pathway of creating a model that would predict luminal A. You just need two genes, and as I said, algorithm selected them, and based on these two genes and these two cutoff values, you can accurately predict luminal A. Another thing that can be interpreted here is that these two genes are associated with these subtypes. For example, normal, luminal A, or luminal B. So when you create a model like this using supervised machine learning, it's called a trained model. One of the most important aspects in today's data science is that all models you create, all analysis you do, it must be validated. You must be very accurate, right? So you don't want to create some model that is a product of randomness, and then when you come across another data set or another analysis, it doesn't work in the same way. So they have to be standardized, they have to be uh, validated, and trained models are validated in different parts of the algorithm, in different parts of methodology that is used in that algorithm. So model is validated first based on the data set itself. So data set is divided in train and test partitions. So one partition is used to train the model, create a model like this, and then it's tested in another partition to see how good it behaves. There's also internal validation. You don't even have to separate data set in these partitions. And there is also internal validation where this model is created and then it's tested on the same data set to see how good it performs. So a lot of it is in the code, but as I said, it must be validated on other parts of the data or other data sets. And that is a very important aspect of supervised machine learning because we have a lot of external data, and that external data might not be 100% accurate always. So this is another way of uh, analyzing that external data that we have. If we can create good prediction models based on our genes, and if we have enough genes and their expressions in the data set, if everything kind of comes together, if everything fits in, then we will create very accurate trained model. And we will have accuracies like, I don't know, 95, 96%, etc. We will talk about this later more. So such a model uh, could be brought to more complexity. This is another form of validating the model even more. You don't create just one tree, you create more trees. You probably must be asking yourself right now, how do we create more trees? Do we need more data sets? Well, that is one way. You could have a large number of data sets, and then you create a decision model for each of those data sets, and then you test them on other data sets, right? So you cross-validate. But very often we don't have more samples. Sometimes we have just one sample, right? One data set with, for example, 50 samples, but it's just one data set. So how would we create a validated model from just one data set? There is a method that's called bootstrapping. Bootstrapping will be expanded in one of machine learning courses on edu TB platform. For now, we'll speak just about some basic terms. So bootstrapping is actually you take one data set and then you assume it comes from a bigger data set, right? So if you take something from a bigger data set, that's just part of it, right? So you might come up with one result. For example, one mean of the data, right? One mean of some gene. But then you imagine that you have multiple parts of that larger data. So when taking each of them, you will come across with slightly different results, right? Every time you take a smaller part of data from some bigger data set, you might come up with some slightly different results. And from those slightly different results, you create some confidence interval, right? And within that interval, all of your data sets should be contained. So what bootstrapping does is it takes your sample, no matter how big it is, and it assumes that it's a part of some 100 times bigger sample, for example. And then it accounts that there should be some confidence interval and your data is the center. So it assigns small variability in the data to match with that confidence interval. So in other terms, you are creating a large number of data sets from one data set, and they're all slightly different. In this way, we are replicating our data set, and we are creating decision trees of all those data sets. In the end, we are creating a final decision tree, a final predictive classification model that is based on the majority of those decision trees. So in this way, we are validating the model using one of the algorithms that is called random forest algorithm. Essentially, it just creates forest of those trees. So we create multiple trees, but as I said, first bootstrapping must be performed in the algorithm, and then we create more of these trees and create a final validated model out. So this would be a theoretical basis of the analysis that we'll be doing today. We'll use the same data set from ductal carcinoma in situ samples. You have the name of the publication here, molecular portrait of high-grade ductal carcinoma in situ. So it's still same gene expressions, it's the same data set we used last time. I just made some small changes to create a data set that is used for supervised analysis instead of unsupervised, simply by adding that class column with names of those classes. As we said, we have normal breast epithelial samples, and then we have luminal A, luminal B, AGR2, and basal DCIs. We also said we have more than 22,000 genes. So if you open the data set, I hope all of you already downloaded it, and you can open it right now. If some can't access it, okay, and I will. Can you all access the data? If someone can't access the data, we can post more links in the chat for you. Okay, thank you. So, open the data in Notepad, it will look like this. It's a tab delimited file, and it has ID and class column. That is very important to have this class column, because now we are creating supervised data set out of it. It's supervised analysis, but this is the data set that is appropriate for such analysis. So there is another section on the data mining part of the interface. We use that unsupervised section. before we use utilities. Now we have this supervised analysis section. If you go there, you will encounter different algorithms. Sorry. Uh, you will encounter different algorithms that can be used. First, you need to upload data. So, as we said, we want to validate the model that is created. And first thing we want to do is create train and test groups. So I uploaded two files for you. One of them is DCIS train, and it contains most of the data from the initial days. You also have test group or DCIS test txt file, which contains few of those samples, right? So we just want to see how good is our trained model and how it behaves on that test model. So you just need to upload files, and you will go to this group selection button. After that, you go to predefined options, and you go to that uploaded files section, and you just drag those files to test and train group with the corresponding names. So if some of you are doing the analysis right now, you can do these steps. It's very easy. Then we will create this pipeline, and we will get the outputs of which we'll talk about later. Now I will share my screen and show you how to do all this. So, okay, so you go to server t by info, right? And under areas of analysis, you have data mining section. So you go to supervised analysis, and you upload the files. As we said, these are the two files. You might come up with this error. As we said, you just reload the website, and you click start here, and then you upload files again. So the two files will upload, and then you go to group selection. You have option to use this drag method to just drag those files in the groups. So train goes to train, test goes to train test group, and then you go to continue. So after clicking start, you will have some explanation of what supervised analysis is and what options you have, what are classifiers, what is classification, what files do we use here, and I strongly recommend that you read this. As you can see, we are mostly using these label data and txt files. So as you can see here, test data can be unlabeled. So we're testing the model we created on label data, and as I said, you don't need to have a labeled test files. So here are the algorithms. As you can see, there are some various variations of random forest algorithms. You have LDA, QDA, decision tree. You have that algorithm we said before was universal. So we had this one in unsupervised section. We also have it here. So naive bias can be used in both sections. We also have t-SNE here that can be used as an addition to these algorithms to visualize data in a better way. But for now, let's stick to decision tree, the algorithm that we were talking about during the session. If we click decision tree, we just click save and end the pipeline. So you can name your pipeline, for example, decision tree, DCIS. Okay, we already have DCIS. So you finish the pipeline and you run. It should take, as I said, 2 to 3 minutes for the pipeline to actually finish, and you will get those output files. So you can check the progress, as we said here. You can see that it started and it's going to run for a couple of minutes. But I've already created same pipeline. It's this one, decision tree DCIS. So it's the same pipeline with same files, and this is the output that you will get. You have just two files, and if you open just one of these two files, it will take you to zip folders. So this one is a zip folder for RStudio. We'll use this one later. For now, we'll stick with this all results zip file. If you open, you will have multiple files within that file. So you have train, test, stat, and you have the plot. First, let's review the plot. If we open the plot, we get that same decision set, right? So these are not three genes, but just two genes that were selected by the algorithm to be best predictors of those four subtypes of DCIS. So you can see this first one, ENSG. It ends on 195 14. This dot 14 should be disregarded. It's just part of some labeling that we use when creating the data set. So this is actually the gene, right? This is the name of the gene. And if you took this name of the gene and went through Ensemble or NCBI, you would find some relevant information about it. I have that information here for you. So that gene is called PIMRAG, and it's actually mitotic regulator. You'll know that mitosis is a process that is necessary for cell division, and cell divisions are closely related to proliferation. And cancers proliferate at high level, and the main problem with cancers is that their proliferation is uncontrolled. So here we identified a mitotic regulator that is related to these four subtypes of, or three subtypes of DCIS. You can see that one of them is normal. Now, what is missing here is basal. So we were not able to create a predictive model for basal, and we'll see later why. So our predictive model is able to predict luminal A, luminal B, and all of them normal, which is for now. So we also have this gene. You can see it's same gene on both of these branches, but it has different cutoff value. So if cutoff value is lower than this, then you can go towards normal or towards luminal. So it's very simple. Just, I don't know, let's use an example. This gene is, it's higher than 0.34. We go to this branch, and then we come this gene, LCO1132, and non-coding RNA. So don't confuse this with non-coding, but it's non-protein coding RNA, and located on chromosome 1. So we can also use related DCIS samples. So if we come across that gene, and for example, value of expression is higher than 0.225, then we have AGR2 sample. So this model could be used to predict any value. You could get a sample, and just looking at these two genes and their values, you can predict with high accuracy which one of these are we talking about. So another important feature, and I'm just repeating this a few times because it's very important, we created the predictive model, but more importantly, we selected two genes associated, and for you, along with this one, to differentiate cancerous cells from normal, right? So if these two genes are lower than 0.34 and 0.135, or with this statistical path that leads us towards normal. This doesn't mean that in real life it would be 100% accurate, but it would be very close to that if you create a good model. So we must differentiate between real-life predictive model, which is associated with, for example, biological pathways, and statistical predictive model. So statistical predictive model gives you ability that something is associated with something. We also created those networks of gene association, and we said that statistical association, but the real point of everything is to create models that are related to biological models. So uh, next step in the analysis of this, we created a predictive model. We saw that these genes are indeed associated with cell proliferation, and they definitely could be related to this BCIS. So next step would be to analyze, for example, these genes in lab. I'm sure there are many publications and other research types that definitely use these genes in the analysis. So we will stop here with this part. So let me get back here. We also have some more files, right, in that zip folder. We had that plot, and now we have some statistics here. Let's, for example, open this file. First, let's extract all of these files. I will just create a folder right here. So I will call it, for example, decision tree, right? And let's extract those files to decision tree. Okay, so you can see they're there, and let's open them from our studio, for example. So we said that we use this part of the interface to upload the files, or to just go to directories. And we always want to set this as working directory because we'll be working with that one. And let's open this one first. Predict dash stat, right? So you can see that it actually creates a confusion matrix. What is confusion matrix? Confusion matrix tell us what is the relation between true samples. We know they're true based on the classification we have, and we also created a predictive model but disregarded that classification to see if the classification is good. So that is one form of internal validation of the algorithm. You have classification at the beginning, right? But then you create a trained model and disregard that classification and use just the model to create the same classification. If they match, they will match in a way that they will be marked in this matrix. So for example, basal and basal, predicted and true, we have value of 0. So they don't match. We'll get back to this later. For now, let's go, for example, to HER2. We have 6 here, right? So they definitely match. 6 HER2 were predicted, but we had just 3 HER2s at the beginning. So what that means is those 3 that were supposed to be predicted were definitely predicted, but it predicted even more, and we'll see later why. Luminal A, we had 7 samples at the beginning and 7 were predicted. Luminal B, 7 at the beginning, 7 predicted. Normal 8 at the beginning, 8 predicted. So pretty much everything was predicted well. Let me just get back here. So everything was predicted well here. Perfect. Even those HER2s were predicted as HER2. But what happened is that 3 basals that were supposed to be predicted were not predicted as basal, but rather as HER2. That's why we have 3 more here. So in essence, overall accuracy of our model is something a bit more than 90%. That 10% is generally these three basals that were not be able to uh, that we were not able to classify. So we classified 8, 7, 7, and 3, not 6 here, but these three basal were not classified well. In essence, we have 90% accuracy in our overall training set. But as I said, when you create a model, you must deal with this inaccuracy. We had this one layer of validation here. And how would we use that layer? Well, simply by disregarding the basal. We definitely can't predict basal based on this model. So this model cannot predict basal, which is actually seen even here because it doesn't appear anywhere in the decision tree. So if you wanted to predict sample that has a lot of uh, basal subtypes, you definitely don't want to use this model. But if you had a lot of samples that, for example, has majority of them as normal luminal A, luminal B, HER2, you would have nearly 100% accuracy on those, right? So you would miss out on anyone that was supposed to be predicted. So this is the first human reasoning part that must be included in this machine learning analysis. When you look at the confusion matrix, you must know that basals will be hard to predict and will probably be able to predict all others in a good way. So we have more files. We have uh, train.txt, which actually tells you how it predicted them. So each sample has a class assigned to it, and then we have this this test file, right? So test file, I test files should not contain classes, but I intentionally let those classes for you to see how good it behaved. So in the txt file that you have as a data set, DCIS test, you have these four samples. And these two are normal, so they're definitely predicted well. This one is luminal B in the initial file and it's predicted well. The only mistake that the algorithm made is AGR2. We already said that our algorithm is predicting basals as AGRs, and that's what happened here. One basal was predicted as AGR. So when you know information on how your algorithm behaves, you could definitely say, hey, these are definitely well predicted, but I'm not sure about this one, right? So you would run the analysis again using some other approach to try to classify
Sorry,
this
sorry, Darko.
one.
I'm
Yes.
sorry to interrupt, but there's a few questions here in the chat.
Okay, thank you. Thank you,
Maybe
Elia.
you could just pause for a second and maybe address a couple of them.
Yes, definitely. We'll pause right here and look at the questions that you get. So first we have the question from Ambi. What type of cutoff value? What actually it means? So I'll just get back here, and you can see those cutoff values. So you have values of gene expressions in your data set, right? So if that value of this gene is lower than 0.34, that means that we're going towards this branch. So that is the cutoff value. It actually tells us what is the decision boundary to go one way or the other. And they're also defined by the algorithm by starting at some 0 point and then going to the maximum and seeing what is the highest probability of having some value as a good decision boundary. So it's very simple. They're defined by the algorithm as the decision boundaries to go one way or the other. Okay, so yes, thank you, Elia. It's actually a threshold of making the decision. So, Ham, the R studio is saying that it's impossible to confusion, there is no value line 1. Also in the plot for matrix. Okay, so okay, so the error could arise from many types of the problems, but generally, if you have these files, you shouldn't open the RPath here, right? So you open the all results. What I did is I didn't call the code from RStudio. I just opened the txt file for you to see it here, right? So this is just a txt file, right? It's not the code. We'll use rcall later in other sessions of the program. But right now I just opened this txt file. So take a look at the txt file. Try to open it in Notepad if it doesn't work, but you should be able to open it in RStudio as well. So
Excuse me, sir. Can I say something?
yes.
Sir, actually, exactly in this procedure, I have downloaded the data. Also, I have downloaded the data from the zip file, and the file named just what you are showing now. I was trying to plot this matrix from this file also, but the thing is there is an error in the RStudio. Also, I have clicked a picture of this, so I can send you later.
Okay, I pretty much get the point. Where is the problem? So we are not using RStudio to create the plots or do anything, right? Right now, we are downloading the files directly from the pipeline. So if you run the pipeline, here is my pipeline, you actually have these two files. And if you go here, you will have all those files in the zip. So we're not going to you don't need RStudio. I can just close RStudio here. So maybe that is the confusion that is created. So you just need to run the pipeline. I will just go back to the pipeline again. Maybe the issues could be there. So if you upload files in a good way, and this is one of the errors that could occur. If you put the files in different sections, for example, if you put the test file here, it won't work. It won't create good output. So make sure to put test here and train here. So just run the analysis, and you can see there are no settings here. So you just go to save and finish the pipeline. So we're not using R now. I was just using R to open the text, right? Because for me it was open and it was easier than opening Excel in that moment. But you can open it in Notepad or any other program. It's just a txt file. So you get to run the pipeline and it will finish. So you will get this output. And you can open this in any program. Try to open it in Notepad, for example. Try to open this one just in Notepad. You also have the overall accuracy, and you also have that confusion matrix. It's just that it's horizontal, right? So it also tells you that Basel has all zeros and it's not predicted well, and you also have this information about others, but it's horizontal. That's why I use R, because it creates a real confusion matrix, but you can also use this one. And you can use it on all of these as well. So you can see that each SRR is assigned with a class, and that is the product of that decision tree. You also have the plot. So the plot is also opened as a PDF file. We're not opening it in R for now. So try to do this, guys. Just run the pipeline again and make sure that those two files are put in good sections. In the train is put in the train section, and test is put test section. So try to do that and let me know in the chat or unmute yourself to see if it's working. Okay, I see from Sohan that it's working. Generally, okay. Let me see. Sarah, is it working for you? Vamshi, okay. How do we consider a model to be good or best model? There is no such thing as good or best model, right? So you could have first impression that this is a good model, right? It's accurate 90%, but it doesn't have to be the case. It has accuracy of 90% internal accuracy, right? So when you test it on itself, it's 90% accurate. It just can't predict basals. But if you tested this on a different data set, maybe it would have accuracy of 60 or 70%. So it's very relative. A good way to create the best model is actually to do what we're going to do next. And I explained that decision trees could be turned into random forest by bootstrapping and dealing with that larger sample and replicates of samples and everything else. So let's do the same thing, okay? I see that Sarah has also fixed the problem. Definitely, the problem is that I opened files in R, but you can open them anywhere, right? I opened them just to review them, right? We didn't actually do any of the analysis in R at this point. So if you upload those two same files, we'll do this quickly. I see we don't have much time, but I think this is very important. Maybe we'll go 5 or 10 minutes over the border, but this is very important. So you upload those two files, right? You do the same thing. You just put them in right sections and click continue. So now you can create a random forest model. Just go here to random forest. And this is high number of trees, right? If we have 22,000 genes, 50 trees would be too much. This is kind of approximation that you get from experience, right? You have to do a lot of random forest analysis to get this experience. But if some of you use this algorithm a lot, you will get this yourself. So I know that this is too much. Let's set it to 100. If you have 22,000, 100 would be something that will definitely produce a result for you. It could be over 200, but let's use 100. Let's use 100 for now. So you just create the pipeline, do the same thing. I'm not going to create the name. I'm just going to run the pipeline. So you will see that pipeline will finish in a couple of minutes, but again, I will use the pipeline I created before the session. You should get the same output. Please give me feedback if you got the same output. Now you have 5 files. Okay, I'm checking the chat, so if some of you don't get the same output, please let me know in the chat. So you have same files here, but if we go to all results here, we don't get to any plot, right? We get the same confusion matrix. We can open it. We have feature importance, and this is also kind of confusing if you open it in Notepad. This is the point where we're going to use R, right? So we definitely need R to open this file. So you go to RPath and you unpack these files. I'm gonna use that same folder, decision tree. I'm just gonna delete this. So sorry. So you just unpack all these files to a specified folder. Now I'm using the same folder, but you can create another one. So when I open RStudio, you will see that I need to actually call the code to create the plot. So let's go to the same directory. Now pay attention to the steps, right? So a small mistake here could just make the code not run well. So pay attention. We have the folder, right? Decision tree. Don't forget this one, set as working directory. You have the R call here. We did this many times on the clustering analysis. So you just copy the code and go to terminal and just paste. So let's see what happens. So you should wait a few moments, right? You have this stop sign here, which tells you that some process is being performed, right? So you shouldn't really interrupt this. You just wait for it to finish. You can see here in the terminal exactly what parts of the code are being run. So now main computations are being performed, classifiers are being trained. That is the part where those decision trees are being created, but not just one decision tree, but 100 decision trees. And we will derive a final model from 100 decision trees. So you just have to wait. This is the part where actual random forest algorithm is being run. You can see the last line here, and it actually takes most of the time. So the whole process could take a few minutes. Uh, we're gonna get back here, but I actually have the output that we will get. So this is the output that we will get. You will get a plot with two files. So one file looks like this and another one like this. Why is this very important? Because we used 100 decision trees now. So we have a much better model than the one we had here, right? So we identified just two big genes. It was pretty accurate, but I'm not sure how good would it behave on some other data set. Because as I said, it's not validated using bootstrapping. This one is cross-validated using bootstrapping. So one sample is kind of replicated with small variability into 100 samples, and then we created predictive model on them. You can see here that as number of trees grows, actually the error is decreasing. So the most accurate models are here when you use over 80 trees. You can see that then it's kind of flatlining, so it doesn't change much after that. So you have a second part. Let me just see if you know it's finished here. No, it's still running, but it's gonna finish at the end of the session, and we'll get back to it. So the second part of the PDF file, it's here, so it appeared. The analysis finished, and you have the PDF file. Let me just open it. So this is what we were actually looking at. So you have two parts of that PDF file. One of them is this chart that shows us how error is decreasing when we increase number of trees. Most important thing of today's session, and that's why I wanted to finish here, is that we use this validated model, and this is most important. Whether you're working for some company as a researcher, data scientist, whether you're performing your own research, whether it's academic or industry, it doesn't matter. The most important thing is to have a validated model. So first model was not validated, it was just internally validated, but this one is validated using 100 decision trees. So if we use this model on some other data set, there is high probability that it would perform in the same way. And that is one of the most important things when doing data science. You must have validated models. You must have models that could be applied on other data sets. So let's take a look at this right part. The left part tells you about mean decrease in accuracy. It could be used to select genes as well. So this is another method. And this is another method of selecting. It says mean decrease in gene. Let's focus on this one since we explained it already. So mean decrease Gini. We said that Gini is actually inaccuracy in prediction. It causes inequality at trend line. So if Gini is high, our predictor is bad. So there is a value that is called a decrease in Gini or mean decrease in Gini. The genes that have highest mean decrease in Gini, means of these decreases actually are considered to be best predictors. So random forest algorithm will select a variety of genes based on mean decrease in Gini. The higher the mean decrease in Gini, the better the predictor. So these are like maybe 20 most important genes for those DCIS cancers. So this is the most important part of supervised analysis we performed today. First, we created some basic decision tree models, some predictive models. We we selected two genes, which are also called features in data science. But now we created this validated model called the random forest, and we selected more genes. We selected 20 genes that could be relevant features for those classes of DCIS. So see, based on this mean decrease in Gini, that there is some hierarchy of importance. First ones are most important, and then it lowers down. But all of these are important. This is statistical importance. I'm repeating this. If we got these 20 genes, that doesn't mean that this one maybe is less important than this one. All of them could be important. So doing more analysis, using more machine learning or data science, and combining it with biomedical data, clinical data, or biochemical analysis would actually confirm how important are these genes. But this is a very good starting point. You have 20 genes that are statistically relevant to those DCIs. So we will conclude today's lesson here. Let me just see in the chat. Okay, so the issue is resolved for Sarah as well. Great. So let me just get back here, because this is one of the before I let you ask questions. Whenever we come across some mistake, it's very important to fix it, right? So if there is something that is not good, always ask. You were probably, some of you were either running the code or using import data set here or some other feature that actually produced errors. What I did at the beginning, right now we call the code. We use this Darko, we use the terminal, right? And we run the whole code and we created this plot. What I did at the beginning, where those errors occurred, is actually this. I just clicked on this txt file, right? So it gives me that confusion matrix. As you can see, we have another confusion matrix here. So some of you might have seen this, right? Class error, but this is not an error in coding. This is just a txt file, right? What we produced as an output is this plot, and we needed to call it through the terminal here. So we did it by copying this code, setting working directory here, and then calling it from the terminal. So right now I will let you ask questions. You can unmute the microphone or you can post questions in the chat. I know that there may be a few questions because we covered a lot of things here, so feel free to ask. Okay, you can ask anytime if you want. So right now I'll just get back to the finishing part of the presentation. In our next session, we'll be analyzing discriminant analysis. So it's called discriminant analysis and classification, but you will see that what we did today was actually similar things, right? So we were creating some classification based on those predictions, right? We were also creating some discriminant boundaries, right? So in essence, we did a lot of those things today, but there is a section of machine learning that is called discriminant analysis, and it actually includes LDA, QDA, and support vector machines. There is this section of machine learning that is called deep learning, and we'll talk about it a little bit in our next session as well. Let me just see the questions. Okay, so no questions. Another thing that I will just mention, and then we'll finish the session, is that a good thing would be for you to try each of these. Sometimes you will come up with an error. Sometimes pipeline won't run or our code won't run. So if you encounter any problems, it's very important to resolve them. This is the best way to actually learn. When you encounter a problem, you resolve it, and that's the best way to learn. You need to do all of this practically in order to have hands-on knowledge, and I see that a lot of you are actually running the pipelines either aside from lessons, either on the lesson as today, and you saw that when we encounter with a problem, we can resolve it, and that's actually the best way to learn. So right now I will finish with the class. Please feel free if you have any questions. Either I will leave my email here again. You can send it also to other parts of the organization or other members of the team. So, okay, Sarah, it's giving me an error when I paste the code. Can you just tell me what the error says? What is the error? Just open, Well, Sarah, can you do this? Just go to that directory where you extracted the files. So make sure that the files are there. Okay, so you open that directory. You should have the files there. And one of those files should be this one, random forest core input.r, right? So don't forget this. Set as a working directory. A typical error that is produced, typical error that you mentioned is when you don't set working directory. So try to do this. Go to the directory, set it as a working directory, then go to terminal and create a new terminal here. Just left click on the terminal and new terminal. And new terminal should contain this path of the directory you specified. Now try to do the same thing, just copy the code and run it here. And tell me what happens when you run it. You will probably have to wait like, I don't know, 5 or 6 minutes as you saw for everything to finish, but you have my mail, just copy my mail and please let me know it should work correctly when you do this. So please notify me on email, did everything run correctly? I believe it would run correctly now. Any more questions? Okay. Thank you, everyone. So see you on October the 17th, same time, and we'll be talking about discriminant analysis and classification. Thank you, everyone. Thank you, team, and see you Wednesday.